Good afternoon, guys. Today we'll start with week 10 of the course Host Pathogen Interaction, that is Immunology by Professor Himanshu Kumar from IISER Bhopal. In this lecture, we'll be discussing about basically the virus body interaction and how the virus infects the human body. And we'll be talking about three viruses here: influenza, dengue, and the Zika virus. Okay, I am Rishabh, and we'll start the lecture for today. Now, moving on, in the last lecture, we discussed about the influenza virus. So, just to give about the brief from the last lecture, it's a negative sense virus that have a spherical to ovoid size and has a segmented genome of about 13.6 kb. So there are 8 segments of the genome, and as you can see here, this is the capsid that have two major enzymes, neuroamidase and the hemoagglutinin, that helps in the basic basic infection of the virus to the host cell. Okay, and these are the 8 segments of the genome that encodes 8 different proteins from the different that perform different functions that we have discussed in detail in the last class. Now we'll move on to the main protein, that is hemagglutinin, that helps in the infection of the virus to the cell. Okay, so the hemagglutinin main function is receptor binding and the fusion activities. Okay, so what happens is, for example, this is the cell, this is the receptor that to which the influenza virus acts. Now, the hemagglutinin basically binds to it, and it helps in the binding of the virus to the receptor, as well as when it enters into the cell, there is a process called as fusion that helps in release of the constituents that is inside the cell into the cytoplasm. So that is called as a fusion event, and those fusion events are actually being carried out by the hemagglutinin also. But there may be a structural role for the HA in budding and the particle formation. The HA is a trimeric rod-shaped molecule, so it is a trimeric molecule that is a rod shape. Okay, for example, for this is the structure of the HA molecule. Okay, so it has a three protein chains or the three peptide chains. That's why it is called as trimeric. And the other enzyme, that is neuroamidase, is a tetrameric protein. Okay. Now the carboxy terminal is inserted into the viral membrane, and the hydrophilic and projecting as a spike away from the viral surface. Okay. So this is the hydrophobic membrane. This is the C terminal. Okay. The C terminal is towards the cytoplasm or the inner of the viral particle, and this membrane consists the hydrophobic part, and the end terminal, that is the hydrophilic, is protruding out of the viral membrane. So this is how the HA protein is organized on the viral membrane. Now what happens is, this viral protein, that HA, is basically different in different serotypes of the virus. Okay, so what happens, there are different types of influenza virus that are being recognized, and this modification is done in the HA protein itself, so that makes the each virus different. Okay, so the cleavage sites present in HA vary between viral strains and can affect tissue tropism, virus spread, and the pathogenicity. Okay, so that's why there is a difference in the viral strains, different influenza strains that we see, that can affect the tissue tropism. That means the type of tissue that they are infecting. Okay, then the virus spread, how easily that particular virus spreads in the body, and it causes its pathogenicity. The highly pathogenic avian influenza virus, that is HPA1V, of subtypes H5 and H7, contain a multi-basic cleavage site that have a consensus sequence of R, X, R. Okay, So this is arginine, then an amino acid, then an arginine. Okay, so this is a consensus sequence of different viral strains, which is cleaved by ubiquitous proteases like furin or PC5 or 6, supporting systemic infection with an often fatal outcome. Okay, so this is how the difference in H is is reflected in different types of viruses. Okay, then there is another virus, that is human airway trypsin like protease and PRS52. This is a transmembrane protease serine S1 member 2 known as epithalicin. This is also present in human airway epithelial cells, and it cleaves HA having a monobasic cleavage site. Okay, so why this cleavage site is actually necessary? Because when the HA is present on this particular membrane, so it requires a form of cleavage. This cleavage actually tend to make it active. Okay, so this is how this HA cleavage takes place. And the proteases that are present in the body, like furin PC56 or HAT or this TMPRS52, these are actually the proteases that help in the cleavage of this HA protein. So this is the basic first step of their infection. Okay, so this is how these HA molecules actually, these virus molecules actually start their infection. Okay, now let us see briefly how the HA structure looks like. So as you can see here, this is helix A, helix B, and this is the fusion peptide here. Okay, and there are different antigenic sites that basically make this receptor or the HA that we are talking about different for different viruses. Okay, and there is another. This is the receptor binding site on the surface of the cells to which this particular virus is going to infect. Okay, now this is the closed conformation in a neutral pH. Okay, now whenever this virus enters, it enters in the form of an endosome. So this is a cell, this is the virus. So when it infects in a form of an endosome. So this endosome has a acidic pH. Now that actually activates this particular HA into an open state that we'll see in the next few slides. So this open state actually helps in activating the fusion event. Okay, now moving on to how this virus actually infects the cell. So in any viral infection, there are four major steps that takes place. Okay, so one is viral attachment, penetration, and uncoating. So the viral vectors actually attaches to the cell, it penetrates the cell, and the uncoating takes place to release the genetic material or the different types of proteins that are present inside the virus into the cell. Then there is transcription and translation of the virus inside the. Then there is the production of the different proteins that are present in the virus, so that the transcription and the translation of the viral genome. Then there is the replication of the virus. Okay, so that it can produce different copies of this particular genome, followed by its maturation and then the release into the system. Okay, so this is how the major pathway for infection or the pathogenesis of the particular virus. Okay, So this is the major, this reflects in all the viral pathogenesis pathway. Okay, now here is the binding of the influenza virus. Okay, so the binding takes place with alpha 2 3 or alpha 2 6 linked sialic acids. So this is the receptor. Okay, alpha 2 3 galactose linked sialic acid or alpha 2 6 galactose linked sialic acid receptors. This is the receptor for the entry of the influenza virus, and these receptors are present at in the respiratory tract. Okay, so they these virus majorly infect the respiratory tract, the superficial cells of the respiratory tract. Okay, and the alveolar macrophages and the dendritic cells can also be infected because they also carry this particular receptor. Okay, so the first thing that decides the viral infection is its receptor and the major site of its organ that is infected. Okay, so the receptors, that is sialic 2,3 galactose alpha receptor and the 2,6 galactose sialic linked sialic acid receptors are basically the site of its entry and the receptor for its infection. The first thing. Then what happens after its binding to the receptor? It is enclosed in an endosome. Okay, so this endosome is basically a, circular uh, process in the membrane that contains the virus after its infection into the cell. Okay, now what happens? So from this endosome, this particular virus needs to release its constituents into the cytoplasm. Okay, so what happens is there is low pH in the endosome. So this low pH basically initiates the fusion of the viral membrane to the endosomal membrane, thereby internalizing the endocytic components into the cytoplasm. Okay, so this low pH actually changes the conformation of the HA, hemoglobin. In the next slide, we will see the whole procedure, and this fusion is therefore this low pH initiates the fusion, followed by release and the uncoating of the virus and release of the constituents into the cytoplasm. Okay, so the internalization mechanisms are four in nature that have been seen in literature. First is the clathrin coated lips, clathrin coated pits, cannuolae, and through non-clathering, non-cannuolae pathways, or through micropinocytosis. Okay, so these are the four procedures through which the internalization can take place. Okay, and the main is the clathrin coated pits. Now, what happens is uncoating of the influenza virus in the endosome is blocked by changes in the pH caused by weak bases like ammonium chloride or chloroquinone or ionophores, example monensins. Effective uncoating is also dependent on the presence of the M2 protein, which has ion channel activity. Okay, so the uncoating of the influenza virus depends on the pH of the endosome. So if this is acidic pH, so the uncoating is basically very good. Now, if the pH becomes basic, for example, after adding the ammonium chloride or chloroquinone, or by the ionophores that disturbs the pH balance, example monensin, so it affects the uncoating. And effective uncoating is also dependent on the presence of the M2 protein that has an ion channel activity. So it plunges in H plus ions so that it makes the pH more negative. Okay, so this is how the uncoating takes place. Now there are some medications that are being approached so that these infection can be prevented. For example, amantadine and rimonabitidine inhibits replication immediately following virus infection. So These both are replication blockers. Okay, so amanitin and rimonabitidine are replication blockers of the influenza virus. Then the neuroaminidase inhibitors are osilotamide and that is Tamiflu and peramivir and zenamivir. So these three are the commercially available anti-influenza inhibitors that affects its uncoating and release of the constituents into the cytoplasm. Okay, so followed by the uncoating, what happens? The RNPs and all the genome that is present inside the particular virus is released into the cytoplasm that enters the nucleus for the transcription and then replication and the production of different proteins. Okay, so the transcription followed by protein synthesis, followed by replication of the viral genome, followed by the process of the production of the different proteins involved in packaging of the virus, followed by its packaging, assembly, and then after assembly, it leads to budding and release of the virus. Okay, so assembly basically takes place with the help of the plasma membrane because it has an envelope. Okay, so the envelope is basically produced by the plasma membrane of the host cell. So this is the whole pathway how it affects and is released from the particular cell. Okay, now we'll see a bit in detail about the fusion process. So as you can see here, this is the closed conformation of the HA protein. Okay, and this is a viral membrane this side, and this is the endosomal membrane, and this is the cytoplasm. Okay, so now what happens? And this side is a low pH. So this low pH water will cause the opening of the particular HA protein. So it opens up so that it can help in. So this is the closed conformation, and this is the open conformation so that it can fuse the membrane of the virus. So this is a viral membrane, and this is the endosomal membrane. So this is fused now, and now the different constituents of the virus, the genome, the RNPs, and different proteins that are involved in the infection are released into the cytoplasm. Okay, so this is the closed and the open state of the particular HA molecule. So this is in the neutral pH. So this is in the neutral pH. This is the closed state. Okay, this is the globular head. So globular head is the, basically detached from the other trimeric molecule that have a fusion peptide. So this fusion peptide help in fusion of the membrane of the endosome and the envelope. Okay, so this is how the two membranes, the endosomal membrane and the viral envelope, are fused with each other. Okay, now moving on to how we can produce it in vitro in vitro conditions. So because now what happens is we cannot produce the intact virus to produce any vaccines or something because of the mere one reason that if we produce an intact virus, it can easily cause the disease into humans. So to prevent that, we have a reverse genetic system in vitro cells like HEK293 and MDCK cell culture, in which what we do is we provide 8 different plasmids that encode for different proteins that are present on the viral capsid. Okay, so this is the hemagglutinin, NP, NS, PB1, neuraminidase, PA, PB2, and M. Okay, so these are the 8 different proteins that are encoded by 8 different segments of the viral genome that are being present in the form of 8 different plasmids. Okay, that have a bidirectional promoter. Now, we'll transfect all these plasmids in a single go into these cells. Now, okay. So now we'll infect these plasmids into the particular cell, and then we'll actually produce those viruses around by 72 hours after transfection. So transfection means that all the 8 plasmids will be produced, will be inoculated with the cells in the form of an transfecting agent. So the transfecting agent can be either polyethanolamine or lipofectamine, based upon the how it can manage into the cells, because this is also toxic to the cells. So based upon what we have to transfect, we use either of these transfecting agents, and we introduce them with the plasmids so that they can easily transfect into the cells. And we are using here HEK293T or MDCK cell culture, and then after 72 hours it will start producing the virus into the media. Okay, so it will release the virus into the media. And these proteins that we have seen here is have a virus name, that is APR8, 34, and H1N. So this is a vaccine step. And this, in similar way, the vaccines are produced for the influenza virus. Now here is some peculiar example where there is a microRNA, that is MIR485, that inhibits the PB1 protein. Okay, so this PB1 protein is a membrane protein, and here MIR485 can actually inhibit this membrane protein that is in use in antiviral immunity inside the cell. Okay, so to test this, what the literature suggests is they have another uh, plasmid that have the uh, MIR 485 uh, MIR 485 construct that can easily inhibit the PB1 protein. Okay, and that they have introduced, and they saw that there is difference in the production of the virus. Okay, so this is how they have seen that the MIR 485 is actually being involved in the inhibition of the PB1 protein. Okay, so this is how we produce the virus in vitro, in vitro conditions, and it can be used to produce different drug targets, then vaccine development, and whether there is any antiviral immune agents that can be used or that can be identified. Okay, so this is the basic function how we can utilize this in vitro system uh, for different functions. Okay, now moving on to the infection part, how it causes the symptoms and the infections. So now we'll see how the influenza virus infects and what is the main path of infection of the and the appearance of symptoms. Okay, so by day one, the virus enters through the body, through mouth or nose membrane, and then it gets infected on day one itself. From day 2 to 4, the virus actually settles in the lungs and it begins to multiply. And because it is contagious, so because this is a communicable disease, so it can infect to the different peoples also. Okay, at day 5, the symptom strikes because the viral load increases in the body. Okay, so fever, headache, muscle, aches, sore throat, cough, and fatigue will come from the day 5. So before day 5, there can be slight increase in the temperature so that you will know whether the infection is actually being taken place or not. Then from day 6 to day 9, the flu hits its stride. That means it increases. During this time span, your immune system of the body is under siege from the virus. Okay. Now from day 10 to 14, your body produces the antibodies needed to destroy the virus. And then at that point of time, we slowly start to recover because our immune system now is being functional to remove the virus from the body. Okay. So now, as we can see here, the symptoms include the fever, headache, tiredness or body aches, then runny and stuffy nose, coughing and sneezing, and the sore throat. So these are the major symptoms what a flu or a cold will look like. Okay. Now these symptoms are majorly caused by rhinoviruses, 50% of the cold. Then majorly it's also caused by the influenza virus and also caused by the coronavirus and the respiratory syncytial virus. Okay, so these are the major viruses that causes these kind of flu and cold like symptoms. Okay, and the flu that we talk about. So this is combination of the symptoms that appear in a flu. So it is an Italian word meaning influence, and flu came from the word influence. Okay, so in case of the viral pneumonia, the infection is asymptomatic. Okay, the symptoms are abrupt, that is headache, chills, and the dry cough, which is rapidly followed by high fever, myalgias, malaise, and anorexia. Okay, so subternal tightness and the soreness can accompany the cough. The prominent site of infection is fever that often peaks 24 hours at 38 degrees Celsius to 40 degrees Celsius. Okay, so after 24 hours after the infection, the fever actually boosts up. Okay, because the body is trying to circumvent that infection, so it increases the temperature of the body from 37 degrees Celsius to 38 or 40 degrees Celsius. Okay. Now, the elder people can have high fever, lassitude, and confusion without respiratory signs. Okay. Conjunctival inflammation and excessive tearing may also occur in case of the eyes. Now, illness is more frequent and more severe in cigarette smokers. Now, in children, if we talk about, for the virus can be found in up to 13 days after the onset of the symptoms. Okay, generally, after the onset of the symptoms, the immune system becomes active. But in case of children, because the immune system is not that active, the virus can be found till 13 days after the infection also. Okay, in immunocompetent patients, the virus that is AH1N1 PDM09 virus is typically shed from a day prior to the onset of symptoms to day 5 to 7 days after the onset of illness. Okay, because they are immunocompetent, so the virus are basically shed before a day prior to the onset of the symptoms. Okay. In school children that has a median age of 8 years, this virus was isolated and median of 2 days after fever resolution. Okay. So after the onset of the fever, after 2 days, this virus was actually being isolated. And there is a drug that is oseltamivir treatment within the 48 hours of symptoms onset significantly reduce the duration of the shedding. Okay, so here the shedding means production of new viruses. Okay, so this drug basically within 48 hours of the symptom onset can decrease the duration of the shedding. So this is how different people have seen the onset of the symptoms and the viral load in that particular age range. Okay, now moving on to the different so this is basically the how the body induces an antiviral response in terms of the interferon and the antibody titers after the virus is infected into the body. Okay, so this is a viral titer. So these are the days after virus administration. At day 2, the virus load is very high in the body, and it reduces till 6 days after the infection. Okay, now how it reduces? Because after the second day, because the load is high, the interferon levels are also high. Okay, so this interferon levels increases up to 3 days, and then after the decline of the viral load, it also decreases till day 8. And as the viral load increases, the production of antibody increases, and it increased to a so there are three components here. First is the nasal wash antibody titer. So in the nasal wash there would be some neutralizing antibodies. So in the nasal wash, there would be some neutralizing antibodies. Okay. So these neutralizing antibodies will go on and increase its load from day 8 to the last 1 week, 1 month. Okay. And then we have the serum anti-neuraminidase antibodies that are somewhat low in titer, and that also increases after day 8 when the virus load is subsided. Okay, and then we have the serum neutralizing antibodies in the last term, that as the lowest titer. Okay, so this is how the different types of antibodies that have been studied are seen in different titers. Okay, so this is for the production of different antiviral immune response inside the body. And it also depends what is the primary host of the virus. Okay, so for example, this H5N1 influenza virus has a primary or a natural host, that is avian birds, and its infection is asymptomatic, and the infection takes place in heart or a muscle, and the immune response that we talk about is low. It has low cytokine levels and a high viral recognition. Okay. Now, if it infects the transmission host through which it is being transmitted to the humans, then they have a mild to severe infection, and the infection is in lung or brain, and they show a somewhat increase in the cytokine production. Now, if they transmit to the terminal or spillover host, there is a severe infection because the terminal host cell, have not seen any kind of infection like that, and it is a systemic spread. So the high level of cytokines, chemokines, and acute phase proteins are seen in this cytokine storm. So, and this is actually very contagious as well as deadly, as we have seen in the Spanish flu in the 90s, in the 60s, 1960s, and now we have recently seen in the coronavirus. Okay, because it's not the primary host of the humans are not primary host of the virus. So, if the virus infects in a non-primary host, basically the terminal host, it will be very deadly in terms of the infection. Okay, the infection basically changes throughout the respiratory tract, but the most clinically important pathology develop in the lower respiratory tract, that is towards the lungs. Okay, acute diffuse inflammation of the larynx, trachea, and the bronchi are observed in mucosal inflammation and edema. Then desquamation occurs. That means infiltration of the neutrophils and the mononuclear cells. Influenza virus specific antigen is present in the alveolar epithelial cells. Then necrotizing changes may occur with rupture of the alveoli and the bronchiole cell walls. Okay, so that means necrosis happens in those particular regions, followed by complete healing of the epithelial damage takes place about till 1 month. In children, the intensity is higher because of the presence of the low immune response. Then, otis, crow, pneumonia, and myostis are more frequent in children than in adults. Influenza A virus has been shown to exaggerate asthma. Children, especially those younger than 3 years, have a higher incidence of gastrointestinal manifestations such as vomiting and abdominal pain also, in addition to the the pulmonary infections, and infection of the neonates can be life threatening. Okay. So this is how the influenza virus can affect the human body and the changes it shows after the infection, mainly the symptoms, the viral load, and the different things. Okay. So now moving on to the questions related to the particular influenza virus that we have studied. Which of the following mechanisms can potentially be targeted to prevent influenza virus replication? So, what can be the correct answer? So, let's see the options. The inhibition. How can we prevent the influenza virus replication? By inhibiting the viral host attachment to the host cell. Yes. Blocking the transcription and translation of the host cell. No. Preventing viral uncoating within the host cell, yes. Then disruption of the host sialic acid synthesis, no. Interfering with RNA replication of the virus, yes, this is correct. Inhibiting neuraminidase activity, yes. Increasing host receptor expression, no. Promoting cytokine storm induction, uh, yes, this can also be correct. So let's see the options. So so blocking of the transcription and translation of the host cell, this can be partially correct because we cannot block the transcription and translation of the host cell in a global scale. Okay, so that's why we have to be targeted in terms of the cells infected by the viruses. Okay, so this is how we see the blocking. Okay, so the correct answers are 1, 2, 3, 5, and 6. Okay, now let's move on to the next question. Which of the following is not a function of the hemagglutinin protein of the influenza virus? What can be the correct answer? So, hemagglutinin basically helps in binding to sialic acid on the host cell. Yes. Membrane fusion, viral genome replication, antigenic recognition, and triggering low pH conditions. Okay, so triggering low pH condition is a yes. So there is no viral genome replication that takes place with the help of hemagglutinin. Okay, so hemagglutinin is a protein present on the surface of the virus that helps in virus binding. The fusion event, then the third one is low pH to basically start the fusion event, and the last one is antigenic recognition. These are the four major functions of the hemagglutinin, but it doesn't help in the viral replication. Okay, now moving on to the next question. Why is low pH important for the influenza virus during the fusion process? So, what can be the correct answer? Okay. So the options are to enhances the virus ability to mutate rapidly. No. It activates the host immune responses against the virus. No. It is necessary for the successful entry of the virus into the host cell. Yes, this can be correct. It promotes the replication of the viral RNA. No. It increases the stability of the viral envelope. No. So the correct answer is option 3. That means that is necessary for the successful entry of the virus into the host cell. So how? Because, for example, this is the cell, and the virus enters into the cell through a uh, endosome. Okay, so now this viral particle is basically now, because its envelope will be fused with the endosomal membrane to release its components inside the cytoplasm. So, this fusion process is mediated by sorry. This fusion process is mediated by the hemagglutinin protein that is mediated by low pH. So low pH changes its conformation that leads to the activation of the fusion peptide, and then it leads to the fusion of the membrane and release of its constituent into the cytoplasm. Okay, so this is the fusion event. Now, moving on to the next question. Which drugs have mechanisms that can prevent viral uncoating and replication in the influenza virus? So, what can be the correct answer? So the viral uncoating basically depends on the pH, and here the chloroquinone and the amantadine basically affects the chloroquinone affects the pH, that affects the viral uncoating, and the amantadine affects the replication of the influenza virus. Okay. So these two have a mechanism to prevent the viral uncoating and replication in the influenza virus. Okay. So now moving on to the next question, which of the following statements is true regarding the influenza virus genome? So what can be the correct answer? So let's see the options. It consists of a single continuous RNA strand. It is a segmented genome of 8 RNA segments. The genome is double stranded DNA. It replicates within the host cytoplasm, and it has a single stranded DNA gene. So the correct answer is option number 2. The genome is distributed into 8 different RNA fragments that encode for different proteins. Okay, so the correct answer is option number 2. So these 8 RNA fragments encode 8 different proteins involved in the replication, and then those are the matrix proteins and the membrane proteins. Okay, including hemagglutinin and neuroamidase. Okay, so this is uh, this is the correct answer. Now moving on to the next topic, that is the arbovirus related diseases. Okay, or the arbovirus related infections. So now moving on to the arbovirus diseases, that is arthropod bone viral diseases, in which the arthropods are basically the vectors that transmit the viruses to the host or the human being. Okay, so the arthropod bone pathogens account for more than 70%, 17% of the infectious diseases, and these are the blood feeding arthropods such as mosquitoes, ticks, fleas, kissing bugs, biting flies, and the lice. They serve as a biological vectors for human pathogens. Blood-sucking arthropods transmit virus from one vertebrate to another viremic host. The virus multiply in the gut tissue of the arthropod without causing any problem to the vector. Okay, so now it actually without causing any problem to the vector, it can replicate inside the gut of the particular arthropod, and it gets replicated there, and now it will infect to the different human. It will be transmitted to different humans. Most arboviral diseases are caused by infected mosquitoes. Several hundreds of arbovirus are known, however, approx 100 viruses are known to cause disease in the humans. Life cycle of viruses are simple, however, sometimes it requires a non-human vertebrate hosts like a bird, horse, etc. in order to transmit virus by a species or a mosquito or a tick. Most of the viruses belong to Tagovirus and Flavivirus family and causes Tagovirus and Flavivirus encephalitis. All Flavivirus transmit through mosquito or ticks except hepatitis C virus. Okay, the most prevalent arbovirus diseases per year are dengue fever, chikungunya, and Zika virus. So these three are the major arbovirus diseases. And then the yellow fever, Japanese encephalitis, and the West Nile virus. Okay, so these three are the least common diseases of all the arbovirus diseases. Then fever with or without benign rash, encephalitis, inflammation of the brain causing high fatality, and hemorrhagic fever, it is quite severe and fatal. So these are major symptoms that are associated with the arboviral infections. So fever with or without benign rashes are the most common symptom, and as it gets severe, encephalitis and the hemorrhagic fever comes into play. Okay, moving on to the general pathogenesis. So they multiply in the myeloid and lymphoid vascular endothelial cells, and if virus actually crosses the blood-brain barrier, then it can multiply in nerve cells of the CNS and may cause encephalitis. Neuroinvasion depends on level of viremia, genetic background, and innate and adaptive immune status of the host and the virulence of the virus. Arbovirus infections show age-dependent susceptibility to CNS. In general, infant and the elder people are susceptible. Incubation period is about 4 to 21 days, and causing flu-like illness to the encephalitis is a severe symptom. Now, some of the individuals also show drowsiness with stuporous, that is completely suspended sense or sensibility, dull or no response. Then mental confusion, involuntary movement, sequelae, personality change, or maybe paralysis. Okay, so these are the different most severe symptoms of those particular uh, patients. Then the general diagnosis is recovery of the virus, direct detection, or serological method. So we can either recover the virus from the fluids or the from the fluid samples from the patient. The direct detection is directly using the primers specific for the viral genome. Okay, so that we can use a direct detection method and through serological methods, so that we can do the general diagnosis of the arboviral based infection. Then immunity against the arbovirus is either permanent, humoral, or the cellular immunity develops after the infection. With age, immunity enhances as proportion of antibody against arbovirus increases, and most of the viruses cross-react among arbovirus as antigenic determinants are related. Okay, so the antibodies that are developed against the different arboviruses are common because most of them are related. And the antigenic determinants are related among them. The treatment includes protective measures, mosquito repellents, and protective clothes. And most of the arboviral diseases are symptom management and not the complete cure for them, because we have to make sure that the virus particle is basically stopped the replication inside the host cell. Okay, now moving on to the dengue fever. So dengue is caused by a dengue virus that belongs to the flavivirus gene and that of the Flaviviridae family. So it is a single stranded positive sense RNA genome that cause full spectrum of clinical manifestation following the dengue virus infection, ranging from asymptomatic infection, dengue fever, and in severe cases it causes dengue hemorrhagic fever and dengue shock syndrome. Okay, so in severe cases it is basically dengue hemorrhagic fever or dengue shock syndrome. The major vector for the dengue fever spread is Aedes aegypti. Is Aedes aegypti. So Aedes aegypti is the main vector for dengue transmission to humans, and in some respect, Aedes albopictus is also there, that can also transmit the virus. About 1 in 4 people gets infected by the dengue will get sick, and 1 in 20 people who get sick develop very severe dengue. And the severe dengue cases can result in shock, internal bleeding, or even death. Okay, so this is how the symptoms appear in case of the dengue. Now, the dengue virus can actually be sy- uh, sylliotic or enzootic or epidemic in nature. Okay, so in case of sylliotic infection, what happens? The primates are basically the budding cycle of the particular mosquitoes. So when the mosquitoes are can infect the different primates, but when these mosquitoes infect a human that carries the virus from the primates, that causes an epidemic. So from one human to the other, it transmits, and it and it is maintained in the human to mosquito, human to human cycle transmission. Okay, so this is how a dengue fever can actually be infected uh, among different living beings in the whole system. Now, uh, we'll see into different. Uh, so, the dengue fever is a febrile illness with two or more of the following: headache, retroorbital pain, myalgia, arthralgia, rash, leukopenia, hemorrhagic manifestations, and virus recovery. So, serological response or temporal occurrence with other cases can also be seen. Now, the so these are the different types of fever or the syndromes that are seen in after infecting with dengue. And the second one is dengue hemorrhagic fever. So it is a 2 to 7 days fever. Then follows the petechiae, bruising, or tourniquet test. It also shows other hemorrhagic manifestations like severe bleeding. Then there is increased vascular permeability, rising hematocrit, hypoproteinemia, serous in effusion. Then we had hypovolemia, weak pulse, hypotension that leads to profound shock. Then decimated intravascular coagulopathy from the thrombocytopenia. Okay, so that actually depends upon different grades of the hemorrhagic fever that we develop. So in grade 1, we have these kind of symptoms. In grade 2, we develop somewhat severe symptoms from the initial symptoms seen. In grade 3, more severe symptoms are seen, and in grade 4, we have the coagulopathy, profound shock, and the severe bleeding in case of the patients that are suffering from the hemorrhagic fever. Then we have the dengue shock syndrome. So dengue shock syndrome is rapid weak pulse, narrow pulse pressure, or the manifestation of hypotension, cold, clammy skin, and the restlessness. Okay, so this is how the dengue shock syndrome takes place. Then the time course of the clinical signs or manifestation is, so these are the days post infection. Okay, so by fourth day, to 10th day, it's the dengue fever. So dengue fever symptoms can profound from the 4th to the 10th day, and rash, fever, headache or pain, or viremia are the four symptoms that are being seen in the dengue fever. And if the severe case is concerned, then the dengue hemorrhagic fever or dengue shock syndrome, the shock, thrombocytopenia, and bruising can develop till 28 days after, and with the rash, fever, headache, pain, and viremia in combination. Okay, so these are the clinical symptoms and how and in what frequency and the timeline they appear in the system. So this is a disease till the 18 days. So from 0 to 10 days, the dengue fever will persist, and after that, after 10 days, the dengue hemorrhagic fever or the dengue shock syndrome will appear. Okay, based on the neutralization assay, four genetically similar but antigenically distinct serotypes of dengue virus identified as Den V1, V2, V3, and V4 are identified. And infection of one serotype confers lifetime immunity against that serotype but not against heterologous serotypes. Secondary infections with a heterologous serotype can lead to severe complications like DHF and DSS due to the process called antibody dependent enhancement. Okay, so as we can see here, these are the antibodies against different dengue viruses, and when it attaches to the Fc gamma receptor, that is the receptor for the antibodies, then to the monocytes, uh, it can engulf the virus and it can increase the production of the virus, so that it can lead to the disease. Okay, so moving on to its infection cycle. So mosquito infection so mosquito basically takes a blood meal from a person that have acute dengue, and then the pathogen is actually incubated inside the mosquito. So then there is extensive incubation. The virus basically infects the midgut and eventually travels to the salivary gland, usually 8 to 10 days. Okay, so the in the midgut, the virus actually multiplies, and this then it transferred to the salivary gland where it is stored, and once the it this vector infects the other human, it can infect it infects and adds the virus to the other human, and then there is intrinsic incubation. The onset of symptoms include 4 to 7 days. Okay, now when the blood meal is taken by another mosquito from the same person or different infected person, then it can again infect it can again incubate inside the mosquito, and it will be a cycle of the dengue virus infection. Okay, the factors that control the dengue virus disease response is the immune status. Okay, so what happens is, if there is more antibody um, receptors on the monocytes that carry the viruses, then there is increased viral load. Then the viral strain, the virulence, neutralization, escape mutants, then the epidemic potential, and the increased viral load basically helps the viral strain to increase the infection. The genetic status in the the genetic status basically shows whether the genetic status allows the person to be protected from the dengue virus or it is susceptible to the dengue virus. Then age, the dengue virus infection increases with age with severity. So with age the severity increases, and the sensitive individuals, that is the smaller age, the infants are also at high risk. So these are the different factors that are involved in control of the dengue virus disease response. And this is where we have to detect the virus during the initial infection. So in the acute illness between the 0 to 6 days, there is increased viremia. So till the fifth day, the viral load decreases, and during that time, we can isolate the virus and do a quantitative PCR so that we can check whether the infection is there or not. So RT-PCR is reverse transcriptase PCR because this is also a viral RNA genome virus. So to basically detect the number of copies of those viruses, we have to do a reverse transcriptase PCR so that we can develop the cDNA copies, that is the complementary DNA copies, and detect it under the PCR conditions. Okay, following the infection, there would be increased antibody titers. Okay, that can be quantified by ELISA. So this is a this ten- or a long-term detection process that can be seen. So we can either detect IgG or IgM post the infection till 50 days. Okay, so because it is a latent infection, and they can store the body can store the memory antibodies for the virus as well. Okay, so there are certain treatments and preventions. So there is no specific treatment that is currently available, and analgesic and antipyretic drugs such as paracetamol are basically given to overcome the symptoms. Fluid therapy is key to dengue management and is applied based on disease severity. Prophylactic platelets transfusion are given for dengue fever with thrombocytopenia to prevent hemorrhagic complications. Bed nets or repellents to avoid mosquito bites, especially in the rainy season and where there is accumulation of water. So we have to prevent that to prevent the multiplication of the vectors so that they will not cause any vector-borne diseases. Okay, we have certain vaccines that are under clinical trials, but they are not being approved yet. So there are live attenuated vaccines. So these are the virus isolates basically, or chimeras, that are produced by different institutes, and these are in phase 2 clinical trials. Okay, then we have subunit vaccines that are basically the proteins on the capsid that are being there. So these are the subunit vaccine against the proteins, and there are inactivated viruses. So the purified inactivated viruses that can be seen in uh, the inactivated viruses that can actually increase the immunity against the viral gene. Okay, now moving on to the other vector-borne disease, that is Zika. Okay. Now moving on to the Zika virus, that is another arbovirus. So Zika is a positive sense RNA flavivirus that also belongs to the Flaviviridae family. So Zika virus was first isolated in 1947 in the Zika forest in Uganda, where the transmission of the ancestral African lineage of the Zika virus was limited to the zootic circulation between non-human primates and the syphilitic Aedes mosquitoes, with sporadic spillover infection to humans. Now, Zika spread mostly by the bite of the infected Aedes species of the mosquitoes, that is either Aedes aegypti or Aedes albopictus. Okay, the mode of infection is mosquitoes first, then the sexual, blood transfusion, or pregnancy. The patient with infection are mostly asymptomatic, 50 to 80%. They have the mild disease in 20 to 50%, and the complicated disease in less than 1%. The severe complications include neurological Guillain-Barré syndrome, acute myelitis, acute transient polyneuritis, and meningoencephalitis. Ocular hypertensive uh, iridocytosis, unilateral acute maculopathy, and bilateral posterior uveitis, coloretinal scars. Then we have thrombocytopenic purpura, transient myocarditis, overall case fatality less than 0.04%, mostly among immune suppressed patients, those with coexisting conditions. Okay, so these are the severe complications that we have seen in the Zika virus case. In case of the pregnancy, how the Zika virus can be transmitted to the baby. So basically, it can be either symptomatic or asymptomatic. Okay, then in 20 to 30% fetuses and neonates that have the infection, There is either fetal loss, that means the baby actually dies in the womb itself, that is 4 to 7%. In the congenital Zika syndrome, there is microcephaly in 4 to 6%, and this occurs in 5 to 14% of the cases. In case of the asymptomatic, medium and long term sequel is observed. And in the rest of the cases where 70 to 80% of the fetuses and neonates that are without infection, they pass out uninfected, and this is seen in 79 to 91 percentage of the uh, 91% of the pregnant female cases. Okay, so that means only 20 to 30% of the people are affected that can be infected by the Zika virus. But the most of the patients go uninfected, and the baby carrying the pregnant female carrying the baby are also safe. Okay, moving on to the different complications of the Zika virus. So first is the Zika associated Guillain-Barre syndrome. So it is estimated to about 2 to 3 cases per 10,000 Zika cases. Okay, so it is a very severe form, and in this, acute inflammatory demyelinating polyneuropathy occurs. So, acute molar axonal neuropathy and the Miller-Fisher syndrome. That is a subset of the Guillain-Barre syndrome characterized by ophthalmoplegia, ataxia, and areflexia have been observed with Zika-associated Guillain-Barre syndrome. Okay, so in this case, what happens? There is a nerve. This is a normal nerve, and this is the demyelinated nerve that has been affected by the Guillain-Barre. Okay, so as you can see here, this is the exposed fiber, and this is a damaged synuclein. So it affects the majorly the neurological symptoms. Okay, so that's why the neuropathy occurs in case of the Guillain-Barre syndrome. Zika associated Guillain Barre syndrome results in higher morbidity and more frequent cranial neuropathy. So, this is a very severe case of the Zika virus where can reach to the brain or the central nervous system, causing demyelinating neuropathy of the particular neurons. Then we have a congenital Zika syndrome also. So the clinical manifestations of are severe microcephaly, collapsed skull, redundant scalp skin. Okay, as you can see here, this is a collapsed skull. There is a redundant scalp skin, hip dislocations. Okay, so the body structure basically is disrupted, developmental delay, visual impairment. As you can see here, the specs are on with a very small age itself. Okay, deafness and body tone abnormalities. So these are some of the clinical manifestations of the congenital Zika syndrome, in which the baby is infected, the mother was infected, and that has been transmitted to the baby. Okay, so these are the basically the MRIs of the skull of the babies. Okay, now moving on to different questions. So, which of the following infections is not transmitted via mosquitoes? So, what can be the correct answer? So, Zika virus is transmitted by the Aedes aegypti. Same with the dengue virus, West Nile virus, and the yellow fever are also transmitted by the mosquito. So, the correct answer is Powassan virus. It is not transmitted by the mosquitoes. Okay, So let's move on to the next question. What is the mechanism by which secondary dengue infections can lead to more severe disease such as dengue hemorrhagic fever? So what can be the correct answer? So increased viral load due to reinfection, antibody dependent enhancement, uh, immune evasion by the virus, cross-reactive T cell response, and activation of the complement system. So the antibody-dependent enhancement is a major cause of the secondary dengue infection. That means, so we have seen in the previous slides also, the antibodies that are against the virus when it infects the monocytes, enters the monocytes. Now monocytes will now be a hub of producing the viral particles. Okay, so now they will produce the viral particles. Okay, so this is how the reinfections of the dengue virus can cause to dengue hemorrhagic fever. Okay, now moving on to the next question: what type of virus is Zika virus classified as? So what can be the correct answer? So it contains the positive sense single stranded RNA genome. Okay, so this is the genome that is present in the Zika virus. Okay, now moving on to the which virus family predominantly included arbovirus responsible for encephalitis. So what can be the correct answer? So the correct answer is option 2, that is Tagorviridae. So Tagorviridae is a class, is a family that contains the viruses that can cause encephalitis, Japanese encephalitis. Okay, so move on to the next question. Which of the following statements about dengue are true? So what can be the correct answer? Okay, so let's see the options. Dengue fever is caused by a virus transmitted exclusively by mosquitoes. Yes, this is correct. Then, infection with one dengue serotype guarantees immunity against the other serotypes as well. Yes, this is also correct. The antibody dependent enhancement can lead to severe manifestations during secondary infections. Yes, this is also correct. Early detection methods include RT-PCR and serological antibody tests. Serological antibody test is basically a late, not a early detection method. Okay, there are currently no effective vaccines available for dengue. Yes, this is also correct. Supportive care is a mainstay or treatment of for dengue fever. Rising global temperatures have no impact on dengue transmission, and community awareness plays a vital role in the dengue prevention and control. So, the correct answers are 1, 3, 4, 6, and 8. Okay, so these are the correct answers. So, this was the last question for today. So, we'll close the session for today. Thank you guys for joining in.